Now we are going to start with dermatophytosis. So dermatophytosis is the most common superficial mycosis affecting your skin, hair and nails. Right. So dermatophytosis is caused by a group of fungi known as dermatophytes. Very convenient. Right. So dermatophytes include three genera. Okay, these are Trichophyton, Microsporum and Epidermophyton. Okay, so trichophyton is not going to spare anything. So it will infect all the three structure, that is your skin, your hair, your nails. But microsporum and epidermophyton are kind of biased. So microsporum does not infect your nails and epidermophyton does not infect your hairs. Okay, so now let's see about the little bit about the pathogenesis of dermatophytes. So this infection, dermatophytosis, is basically contracted from direct contact. Direct contact from whom? So when we come in direct contact with any infected person or animal, or it may be just the infected soil which is containing the fungal spores, then we get the infection, because then we basically can scratch our skin or your scalp, right? And then the infection can occur. So after the direct contact with infected human, animal or soil which is containing the fungal spores. Right. So the lesions that we see in case of dermatophytosis, the one which patients presents with, these are not produced because of the fungal invasion per se. They are produced by the host due to the host inflammatory reaction to the fungal antigens. Okay, so lesions are produced due to host inflammatory reaction to fungal antigens, right? And one more thing, these dermatophytosis are more commonly seen in males as compared to females, so good for us, yay. So females are not commonly infected by these fungi because we have progesterone, right? We have progesterone which is inhibitory to growth of dermatophytes. Okay, so progesterone is inhibitory to the growth of dermatophytes. Now let's talk a little bit about the kind of lesions that we are going to see in the particular cases. So in case of skin, what happens is these fungi are going to grow in a centrifugal pattern, right? In a centrifugal pattern in stratum corneum, and they are going to produce annular or ring shaped lesions with central clearing and raised edges, right? In case of hair, it could be different, because these fungi can either invade the hair shaft. Okay, when they invade the hair shaft, it can lead to alopecia, right? So when they invade hair shaft, alopecia may be present. And if they're just around the hair shaft, they're just surrounding it, there is normally no alopecia, so they can either just surround it, right? In case of nails, they are going to infect it from the lateral side, and then they will cover up the whole nail, onychomycosis. Okay, infection of nail, so they are going to invade from the lateral side or edge of the nail. Okay, so now that we are done with the pathogenesis and the basic intro, let's see the type of dermatophytosis that we normally see. So first one is tinea capitis, which is nothing but the infection of scalp. Okay, so this tinea capitis is again of four types. We have chirion, we have pavis, we have ectothrix and endothrix. So in case of chirion, it is caused by these two fungi, Trichophyton mentagrophytes and Trichophyton verrucosum. And what happens in chirion is there is painful inflammation there's painful inflammation of scalp, which is causing boggy lesions. Okay, there is boggy lesions on scalp. And in case of favus, there is a cup-like lesion, a cup-like lesion, which is also known as scutula. Okay, which is also known as scutula, is formed around the infected hair follicle. Where it is formed? It is formed around the hair follicle. Right. So we saw that chirion is caused by Trichophyton mentagrophytes and Trichophyton verrucosum. And in chirion, we will see painful inflammation along with boggy lesions on scalp. And in case of favus, which is again a type of tinea capitis, there will be a cup-like lesion, which is also known as scutula, around the hair follicle. Now, let's see. Favus is caused by Trichophyton sholani and Trichophyton violatium. So how to remember them? So there is a little bit of mnemonic. So we all have watched MTV in our times. So from T we have Trichophyton, right? From M we have mentagrophytes. From V we have verrucosum. So MTV you can remember for chirion, and in case of favus we can remember it by TVS. TVS is maybe a type of scooty. So from T we have again Trichophyton. From V we have violatium, and from S we have sholani. Right. So the next two type of tinea capitis we have is ectothrix and endothrix. So ectothrix is caused by these three species. We have Microsporum canis, we have Microsporum ordwini, and we have Trichophyton mentagrophytes again. Trichophyton mentagrophytes was causing chirion also. So in ectothrix, we are seeing the arthrospores, which is nothing but the asexual spores, right? So we are seeing the arthrospores which are just surrounding the hair shaft. They are not invading it. Okay, so we see these arthrospores, which are asexual spores, which will surround the hair shaft. So how can we remember these names that I've written? We can not go for MTV and TVS here. I've written it by mistake. Okay, so no MTV and TVS. They were for chirion and favus, right? So from canis, there is, you can always remember your favorite thing, at least my favorite thing, or living person or a living being. You can always remember dog from canis, and from mentagrophytes you can remember men, and from Odwini I have put a picture of Linguini. It was the chef in Ratatouille, so it sounds like Odwini and Linguini. They sound a little bit similar, so I put it in here. So you can think that the dog and the men are going to meet Linguini. Okay, helps me. Now in case of endothrix is caused by Trichophyton tonsurum and Trichophyton violatium. Now in case of endothrix, like I was telling, they are going to invade, right? They are going to invade the hair shaft. Okay, so they are going to invade the hair shaft, and these arthrospores will be totally in the hair shaft. They will covering. They will be filling it completely. Okay, and that is why alopecia is present in these cases. Okay, alopecia is present in these cases. We saw that the arthrospores were just surrounding the hair shaft, but in case of endothrix, they are going to invade the hair shaft. That is why it is resulting in alopecia. Okay, next we have a lot of dermatophytoses. See, we have discussed these already. Okay, tinea capitis, which is the infection of scalp, and under tinea capitis we have discussed about chirion, we have discussed about favus, ectothrix, and endothrix. Right now we have tinea corporis. Corpus means body, so the infection of the non-hairy skin of the body is known as tinea corporis. We have tinea pedis, or the athlete's foot. So it is the infection of the web spaces between the toes, which then spreads to the sole in a mocassin pattern. There is a picture of that, right? Then we have tinea cruris, we have tinea barbi, and tinea unguum. Right, so tinea cruris is nothing but the infection of the inguinal region. Tinea barbi is the infection of the beard, and the tinea unguum is the infection of the nail. Again, onychomycosis. Now let's see about the lab diagnosis. So if a person is being suspected of having dermatophytosis, how are you going to detect it? So again, we have these favorite specimen that we collect from the patient. Okay, in case of in case of superficial mycosis, so we will collect skin scrapings. Okay, we will collect nail clippings. and we can collect hair plugs. So obviously the first step that we want to do is KOH mount because they are keratinized structure. And again, we know the concentration of the KOH used for the specific samples. So if it is skin and if it is hair, we want 10% of KOH. And if it is nail, then we want 20 to 40% of KOH. All right. So after you do the KOH mount, what do you want to look for under the microscope? You want to look for thin, septate, hyaline hyphae. Okay, hyaline hyphae with arthrospores. Okay, so when you do the KOH mount, and if it is a dermatophyte, that is either trichophyton, microsporum, or epidermophyton, you will see a thin septate, not a one, like a lot of thin septate hyaline hyphae with arthrospores. Then we can do culture. Okay, so we do culture on SDA, obviously. Okay, so we do culture on SDA with cyclohexamide, and we incubate it for around 30 days. Okay, for 4 weeks or 30 days at a temperature of 26 to 28 degrees Celsius. Okay, so after these 4 weeks, the growth will appear and it will look somewhat like this. So let me little bit zoom it. Okay, so the first one that we are seeing, this whitish colony, okay, this whitish colony is of Trichophyton pentagrophytes, and this brownish to reddish colony, we have this Microsporum canis, and this one is Epidermophyton flocosum. Epidermophyton flocosum is mainly the only medically important species of Epidermophyton that we have to know. Okay, so next, what else we can do? So after we have done the culture and we have done the KOH mount, we want to know what kind of species or the genera we are talking about, right? So for that, we want to do a LPCB mount. Okay, let me erase this. Okay, so we want to do a LPCB mount, which will help us to see the hyphae along with the spores or conidia. Spores or conidia is the same thing, right? So what does LPCB actually stand for? So we have lactophenol cotton blue mount. Okay, this cotton blue is going to stain the fungi blue, and this is lactate, which is going to preserve the fungi, and phenol is for the disinfection, right? So we want to do a LPCB mount to see the hyphae and the spores so that we can comment on which kind of genera we are dealing with, whether it is Trichophyton, whether it is Microsporum, or it is Epidermophyton, right? So let's see. In case of Trichophyton, so we are going to see two types of spores mainly. We have microconidia and macroconidia. Okay, so in case of Trichophyton, we will see a lot of microconidia. Okay, a lot of microconidia, and macroconidia may be absent, but if they are present, they are going to be very few. Okay, they are absent or they are few, and when they are present, their shape is pencil like. So when they are present, they have pencil shape, so they would look something like this. Okay, then in case of microsporum, we just have to reverse it. Okay, there will be very few microconidia and a lot of macroconidia. Okay, so what will be the shape of macroconidia? They will be fusiform shaped or the spindle shaped. Okay, and in case of Epidermophyton, the microconidia are always absent. You will never see in case of Epidermophyton any kind of microconidia, and the macroconidia are obviously going to be numerous, and when they will be club shaped. They have a specific shape. Okay, they will be club shaped. So let us see these. Okay, so here we are. So let's see. This is the case of microsporum, right? This is microsporum, and we can see the fusiform or the spindle shaped macroconidia. Okay, this is one, this is second, and these are the club shaped, macroconidia in case of epidermophyton, and this is the pencil shaped macroconidia in case of trichophyton, and there are numerous microconidia, right? You can see here there's one another pencil shaped macroconidia, and one is here, one is here. Okay, so now we have done the LPCB mount, and let's say we know what kind of genera we are dealing with. Now what else we can do for the diagnosis? We can do wood lamp examination. Now what is wood lamps? We have already discussed about wood lamp. In case of tinea versicolor, we were seeing the lesions of the patient under wood lamp, right? And they were showing golden yellow fluorescence. So in case of microsporum, the species of microsporum and trichophyton sholini, we also do wood lamp examination. So we have wood lamp examination, and there will be fluorescence of produced due to the pteridine pigment. So fluorescence is produced due to the pteridine pigment present in cell wall of the fungi. And which fungi? We said that it will be in microsporum species, okay, and trichophyton sholini, right? So you have done wood lamp examination also. Then what else we can do? We can do a urease test. If we are suspecting trichophyton, we can do a urease test because trichophyton mentagrophytes is positive for it. Okay, trichophyton mentagrophyte is going to give a positive urease test. We can do a hair perforation test. Okay, and in what happens in hair perforation test is we put the colonies, right, in a petri dish with the yeast extract, right, and with hair. So the fungi are going to pierce. They're going to pierce the hair and produce wedge-shaped perforation, right? And this was all we can do. Some molecular method like PCR can also be done for specific antigens, right? We can do PCR for specific antigen or gene. Okay, we can do specific genes such as chitin synthase. And now that you have diagnosed that it is a dermatophysis case, so what are you going to give to your patient? What kind of treatment you are going to give to your patient? You will prescribe terbinafine or itraconazole. Okay, these are oral drugs. So that was about dermatophytosis, and we are done with superficial mycosis. In the next video, we will start with subcutaneous mycosis.